Proteins, biological molecule proteins. Proteins' basic structure, or the monomer of proteins, are known as amino acids. Here is the basic structure of an amino acid. It consists of an amino group attached to a central alpha carbon, which is then attached to a carboxyl group. Notice the carboxyl group is double bonded to an oxygen and then also bonded to a hydroxyl group. This R molecule here gives all different amino acids their specific properties based on what is on that side chain. You can see I have circled the amine group. I've put a rectangle around the carboxyl group. I've put a square on the alpha carbon, and I have starred the R group. That is the side chain that makes every amino acid unique. There are 20 amino acids. This table actually gives 21. It gives an extra special one here called selenocysteine, but we will not worry about those. Let's start off with the amino acids that have electrically charged side chains. If you look at these, they are either positive or negative. If they're positive, that means they have picked up an extra hydrogen ion, and so they have a positive charge, so they act as bases: arginine, histidine, lysine. Each amino acid has a 3-letter abbreviation as well as 1 letter of the alphabet that describes it. You can see the negatively charged amino acids are aspartic acid, glutamic acid. They're negatively charged because they have donated a hydrogen ion, so they act as an acid because they donate hydrogen ions to the solution. The middle group here of our amino acids are going to be the ones that are polar, uncharged side chains, and you can see they are serine, threonine, asparagine, and glutamine. When you look at them, here is your here is the actual structure, the carboxyl group. This is your central carbon, and here is your side chain over here. Because they have hydroxyls, they can be polar because of the oxygen group in there, or they are polar because of having nitrogens and oxygens, or here's a nitrogen and an oxygen, just a longer side chain. There are 3 special amino acids. They are cysteine, which has a sulfur in its side chain. They are glycine, which is the simplest amino acid. It really doesn't have a side chain. It just has another hydrogen attached on that central carbon. And then the third kind is proline. And proline is unique because it actually the side chain joins back onto one of the amino group here to form this ring structure. The last group of amino acids are the ones that are hydrophobic. Hydrophobic means they do not like to be in water, and there's quite a few of them, and some of them are just longer versions of each other. So here you can see this is just got a carbon, a methyl group on it. This has got 2 methyl groups on it, and you can see these chains of carbon are just getting longer and longer. Methionine, probably one of the most important. The first amino acid in every protein is methionine. Methionine does have an internal sulfur within it. And then we have our ring structure, phenylalanine, tyrosine, and tryptophan, that all have these ring structures as part of their side chain. So those are the amino acids that can be uniquely added into the chain. How we link these amino acids together is known as a peptide bond, and the peptide bonds between the carboxyl end of one amino acid joining to the amino end of another amino acid. Here is showing the peptide bond, how it forms between 2 amino acids. That's actually what a peptide bond would be, and this would be looking like if you're actually looking at the actual atom molecule. Here is showing when you form a peptide bond, it's by dehydration synthesis. This is showing between 2 glycine amino acids, and as you can see how a water molecule is formed between these 2 parts of the amino acid, with a water being given off. Can you circle the next molecule involved in the next peptide bond? You are correct. You can see how that hydroxyl group would form with one of the hydrogens from the amino group of the adjacent amino acid, forming the new peptide bond.